Our next speaker, Mike Gretzinger, is the Farming Smarter Research Coordinator in the Agronomy Program. He coordinates protocols, seeding, spraying, data collection, pictures, harvest, and data processing for the small plot trials and demos. He has been involved with hemp research across Alberta for more than a decade, discovering innovations to enhance the accessibility of the novel crop for irrigation farmers. So we'll welcome Mike to the stage here.
Thank you. Am I good? Kind of forgot what was in my bio there, so I wasn't sure when I was supposed to run up on stage. All right, we've got a good treat for you today. I'm live. I got some sound bites in here too. I couldn't let Ken have all the fun. This is probably also from one of his favorite movies.
Welcome, everyone. I am your damn guide on. Now, I'm about to take you through a fully functioning power plant. So please, no one wander off the damn tour. And please take all the damn pictures you want. Now, are there any damn questions?
Yeah, where can I get some damn bait?
Eddie.
All right, so I thought that was a pretty fitting bite. I'm going to take you through a lot of the industrial side of the grain and fiber processing for hemp. And then I've kind of got the same awful puns throughout my presentation. So what the hemp is Farming Smarter up to. And I'm going to tell you what we're up to here. So we're going to talk quickly about what's going on for grain processing. I actually don't come from an ag background. I took biochem and I did a bunch of lab work. I worked at Black Velvet Distillery doing quality analysis, things like that within the lab, and then also at Maple Leaf Pork through the whole production plant. And so some of this production side stuff is still quite interesting to me, and it's kind of just a little hobby that I have. So we'll talk about the fiber processing. If we get time for grain yields and hybrids, there's some really interesting things going on with hemp there as well. And then if there's some time left over, we'll talk about the new emergence trial and some of the seed treatments and minor use that we're doing there. So this is a little infographic of all the different end uses for hemp. This is just looking at the straw production and the grain production. So as you go left to right, this kind of just breaks down the steps of where we're going. So you'll see up at the top, hemp straw production. We do decortication. We separate that bast and hurd fiber, which I'll show you. The bast goes into things like pulping, into woven materials and other composites. That hurd is used mainly for absorbents and building materials, and hempcrete product. Traditionally not as high of interest in that, but need is growing. Then also on the grain side, we've got the hemp seed, which goes into hemp oils, hemp nuts, hemp cakes, and all sorts of other things there. So it's actually the 4 Fs. I've got my own 4 Fs for hemp here. Fractions, which we're not going to talk about at the top. That's the CBD, pharmaceutical side, THC, and other things. Fiber we will talk about, food we will talk about, and then feedstock. So that's just all the other leftover things that, hey, may as well make some bedding, give them to animals, whatever. So traditionally, we start there on the feed and fiber. But what's interesting, our new project in the hemp side, we get to go on to this decortication side of things. And so we're going to talk more about those industrial processes a little bit today. So I pulled this right off the Manitoba Harvest website. This is their quality page. And so it's a little hard to read, but it goes through some of those quality things that they're looking for. So for example, the seed quality, make sure it's an approved variety. They've got this non-GMO project. That's a big part for their sales side for consumers. They've got a whole safety and traceability program, the GRAS procedures, so generally regarded as safe. They've got certain manufacturing steps that they go through, and then a bunch of other accreditations, so you know you're getting a good quality end product. So what do those things look like? Like if you're like my early days sitting in a lab, well, you need product to come in with low moisture. So this is where our agronomy starts to affect what happens at the processing side. So you need good source to come in. You need an approved variety. So traditionally with Manitoba Harvest, that would be Finola variety. They were taking contracts with RAC, which is a new variety we'll talk about. It's got a bigger kernel weight, and that makes processing a lot easier on their end for some reasons. And then appearance. So we want gray-brown. We don't want any greens in here. So this is something that might have come off a combine or been cleaned through once, but this is what we need the seed to look like. in order for it to be ready on the processing side. It's got to be pure, 99% clean, all the immature stuff removed. We've also got a whole toxin screening, so you have to pass acceptable coliforms and have basically no E. coli present. And then there's a whole grading process as well. This is where some interesting stuff happens. So in the distillery, I got to do like a sensory panel at 7 o'clock every morning. Not as exciting as it sounds at first. And that was to make sure that there weren't off flavors, rancidity, things like that. And so it's just a cool little video I got here. So in this video, he's gonna basically be pressing, these are walnuts, hazelnuts. Extract the oil, centrifuge everything in the lab, and then run it through this machine. As long as everything's under 2 MEQ per kilogram, then they know the rancidity is okay in this case. Press the wrong button. So the sample passed their QA process there. So that's kind of what something looked like on the grain side. Oopsies. I had a pun here. You guys heard that caveat emptor. So this is like a kind of that buyer beware. So this says you don't have to worry about buyer beware anymore. All this testing is going on here. So I've shared this graph before, or this map before. This is listing out all the processing facilities in Alberta. You'll see we're way down there in the south. For some reason, Lethbridge wasn't on this map. I should have added it, but kind of between the Pass, Medicine Hat, and Cardston there. And I updated this the other day and put the Falher one up in blue as well. So this is all the fiber processing facilities in the province. And I think one of the big challenges we have here for fiber in southern Alberta is we just have too many other competing industries. There's, a big McCain expansion. We've got all the other high-valued irrigated crops. And so, to get maximum fiber production, it is nice to grow it under irrigation, which we do in a lot of these cases. And it's just a lot of competition down here in the south. So Jan sent this video. This is their micro decorticator at Initech, Alberta. So we're passing individual stalks through here from some of our trials. I think this was taken a while back where they did a huge analysis on all the variety trials we were doing within the province. You can see the first bit's broken up. There's this big tumbler. So that's where the outer fiber, the bast or bark, is separated and it's spun off and beaten down. And then these first couple collections will separate. So it's important to see how much weight you get within that first pass. They want to know how much fiber goes into each of those tubs. And then this is a good way to separate and kind of start off what we're doing. So you can imagine this is like a micro scale. You could never fit an entire 5,000 acres worth of hemp through something like this. So I went to Canadian Rockies Hemp Corp. And so within their website here, they talk about their processing capability. So their Bruderheim processing is capable of up to 50,000 acres of hemp per year. That's what it was designed to handle. So if you imagine 110,000 tons of herd fiber coming through, I did some quick math here. So 365 days a year, we'll say we work 260 of those days, 8 hours a day, that's 2,080 hours a year. We're probably only 90% efficient. There's going to be downtimes. Everybody's got to have lunch and vacations and stats and stuff too. So just to make the math easy, I kind of went with this 0.9, and that gets us 59 tons an hour. So that's a ton a minute of fiber that's being processed compared to that little micro decorticator putting stalks through. I mean, that's like a bale going through every minute, just constantly over and over and over again. So the thing that I always heard when I was like in any of those production settings is like, don't stop the line. That's the one thing we don't want to do. And so there's a lot of implications for how our agronomy affects that further downstream. So you can imagine $3,000 a minute that you're putting out in outputs there. if you're stopping the line, your CEOs, managers, shareholders, whatever, are not going to be too happy about that. So don't stop the line. Time is money. Maybe I think those ones are pretty generic. You guys haven't heard this one before, so I'm going to make this one up here. Don't ret the bed. That's going to be my new saying for what we're doing with the hemp retting trial here. So hemp retting is something we're going to start talking about and kind of go into our trials and what we're doing. Retting is just rotting. It's just using the bacteria and fungi, moisture to break down the gummy substances and the lignins surrounding those bundles, and it basically allows you to separate that bast and hurd fibers from the plants. There's a couple different methods, and we'll take a quick peek at that here. So this is just showing you the bast fibers. So I just have a scrap of fibers in the back of the truck here. All those long stringy strands on the outside, that's the bast. This interior fibers, the hurd, bast, hurd. So make sure we all get it straight. We'll do this a couple of different times. There's a lot of agronomic things that will influence our fiber production. So this comes from an old paper of work that happened when we were first starting to get into hemp. This is just showing that on the left side there, 300 seeds per meter squared, so really high seeding rates, generally how we seed fiber varieties. That's going to impact the bast to hurd ratio. When we have a low density, like we would with grain type varieties, we're going to have a lot more hurd. So if we think the bast is more valuable and we're using it more on the textile applications and things further downstream, we need that higher seeding rate. And that's just kind of one of the reasons why. Another thing is the varieties. So if we look at different components, varieties can have a huge impact on the amount or the yield that you're actually getting. So within our new study with ARDAR, one of the factors we're looking at is obviously that seeding rate. So this study's focused on looking at the straw management and how that's going to improve hemp fiber quality and productivity. So the first portion where we're looking at the seeding rate, we want to see how that affects things for industrial use. And this is just results from this year. We've got Initech and the Lethbridge site. You can see our tons per hectare is just dry biomass yield. At our higher seeding rate, we got slightly higher biomass production. It wasn't relative to the amount that we seeded. So, obviously we can get away with seeding quite a bit less, just like anything, and still get a relatively good biomass. But where the implication is going to be is when we actually process that and see what kind of quality we get and what kind of ratios we get and that sort of thing. So I think there's more to it always than just that yield picture. And then another thing we're looking at is the harvest timing. So in a fiber crop, generally the optimum yield and optimum quality is going to be at the early onset of flowering. That's like middle of August for our case down in southern Alberta. But if you're growing a dual purpose and you want to get that grain yield from the grain and then go back in and use all the fiber, you're not going to be able to harvest till physiological maturity. So that's going to be closer to mid to late September. In our case, 117 days. So we know that, the longer we let it grow, we're probably going to get a little more fiber. That's all this is showing. But that's also going to have impacts on how long that fiber can then sit in the field and what those fiber quality is going to look like when we get it back into the plant. This is Carlo and I in our little plot swather going across. This would have been at September about 23rd date, just chopping everything down. And then we're going to look back and you'll see, the plots beside us that have been swathed down. Everything just falls down and lays there. Anything that was kind of got tangled up or something, we just laid back nice. And in the foreground there, that's the hemp that would have been swathed at an earlier date. That would have been the mid-August date. So you can see it's already like right brown. It was broken down pretty good at that point already. I guess one of the advantages of that early cutting was that it would have been watered a few more times. We would have had that fall moisture, lots of sun, and all those other things. So what the hemp is retting. What is retting? It's rotting. It's a breakdown, dissolving lignin, gummy tissues, like I said, and other pectins surrounding that bast fiber, and it just allows you to separate it. So we go bacteria plus sun. This is plus the air. Where's my air? And plus dew. That's going to help you get all of your retting. So that's what we're doing. So those microorganisms, I'm going to come back to those in a little bit because I picked those bacteria pictures there on purpose.
Welcome back to the Riverdale Hemp Farm. Today, we are going to examine retting. The word retting is synonymous with the word rotting and is the process of nature decomposing the hemp stalks after they've been cut. This facilitates the separation of the fiber from the stem. Retting requires the action of bacteria and moisture to dissolve away cellular tissues and gummy substances such as lignin. There are two types of retting: field retting and water retting. Field retting, which is common in areas having limited water resources, is most effective in climates with heavy nighttime dews and warm daytime temperatures.
Okay, there's a little more I cut out from there, but there's a bunch of other kinds of retting, chemical retting, enzyme retting, things like that. And so the one thing that we're looking at within this trial is this augmented retting. That's what we're calling it. And so the Inotech crew basically they extracted a bunch of those proteins or those bacteria and fungi that were responsible for the retting process. I don't know the what do you call that? The trademarks or whatever, intellectual property on how they're doing that. But we basically are applying this to try and accelerate the retting process so that early and the later cutting date in the fall, we're going back in and we're augmenting it with additional things, kind of a lot like you'd apply an inoculant to your pulses. We're kind of like trying to speed up this process. And then we've also got a winter retting treatment. So a third of all of our straw is actually still sitting in the field right now, and we're gonna pick it up in the springtime and then take it in and do the testing and see how it goes. Just some shots of the trial. And then this is us bundling things up in the fall. This is a subsample that I grabbed January 31st. This is not in the field for quite a while, but it's nice a few weeks ago. Look how easy this fiber's coming apart. There's just some leftover stuff. I'm pulling this apart with one hand. This is probably over-retted, some of this. Like you see the fibers just falling apart. But further up here, and I found one buried down at the bottom, kind of protected. Pretty tan, and this is, I'm struggling as hard as I can now. Could not get that fibers apart. So if that goes into the facility, nobody's going to be having a good day. If you brought an entire shipment of that kind of thing in, so that's no good. This is just some of the dates. So we planted it May 29th. First timing, first swathing was August 15th, and we applied as soon as we could that augmented treatment. Swath again September 23rd and did an augmented treatment. Picked everything up October 24th, and then anticipating a spring treatment again. So there's going to be, between 30 and 200 days that difference that this hemp has sat in the field. So we're going to have a wide range of results from that. What else do we want to know? There was that graphic I took earlier. There was an entire article or kind of paper published by Inotech. Is like 100 pages long, and this is kind of what I could steal out of there for this presentation. The BAST, we can look at diameter, bundling, and tensile strength. And so that tensile strength is what this Presley fiber bundle tester is doing. They just steal this from the cotton industry, and basically this weight moves along a bit of fiber, and at the point at which the fiber finally breaks, they just measure how far that goes. They come up with this breaking load. They use the sample mass that it took, a couple constants and things like that, and they basically come up with this tensile strength. The other one on the herd side is how much herd do we get through that first pass. So if you have to rerun samples, that's just killing your efficiency. So you want to maximize how much you're getting. So it's a balance between how fast you can go and how much you're getting or losing. So yeah, it's pretty cool. Don't worry, these methods have been empirically tested. So that's the fiber side, and that's pretty cool what's going on there. The next thing that we're working on is some different things with hemp hybrids. So this is all led through Verve Seeds. They were the first company to develop a monoecious female hybrid hemp. So a lot like the canola industry, when it originated, everything was OP. So within a hybrid, male and female are cross-pollinated, and then selectively bred. They want to reduce the dioecious females, and then they want to have as many hybrids as possible to purify their lines. So if we go back, some of you may have seen this before. This is kind of the traditional way we would look at hemp. We've got our dioecious varieties. So a lot of things like Finola would be dioecious, rack would be dioecious, and these are typically for grain production. So if you got half females, half males, those males are just pointless once they've pollinated the flower. The females tend to produce a lot of grain compared to the monoecious varieties over on the right, but they're so ununiform and the plots are so variable. They don't make for good fiber, so they're really only focused on grain. For the monoecious varieties, that's typically more used for the fiber side because you have a lot more consistency. You can grow at that higher seed rate. And then some don't produce necessarily as much grain. And if you don't care, you don't care. If you're trying to dual purpose things, it's nice to pick one that has a lot of grain as well. So within these monoecious varieties, you'll get some that are predominantly male, some that are predominantly female. So this is just showing the two differences. If you have predominantly female, you're probably going to get more seed, which is probably what you're after. And then where things get super interesting with these hybrids is that we'll go here. So we have a hybrid on the left here, and it looks very female. And then within those same crosses, you'll end up with some of these, it looks female, but it's not actually producing any seed. And you end up with this one, it's kind of like, it looks male, but it's not also definitely not going to produce any seed. And if you've got a hybrid crop, you really don't need those male or female looking ones anymore. You just are interested in that hybrid. So the more you can maximize those hybrids, the better off you're going to be. So this is an example of one of our plots from this year. This is one of the OP males. And you can see the difference between that and the hybrid. This hybrid is like 2% other, 98% hybrid. And so on the left side, you can see all the male skeletons sitting there dead, kind of taking up space and probably a lot more gaps between some of those female plants. And then you get some other interesting things. So, in this case, there's the purple bracts. And combining those, it kind of sounds dirty actually now I put up there. So, but this comes from indigo. So there's a reason it's purple there. So it gets the purple from indigo. It gets other traits from the other one that it's crossed with. And so how did things work out in the field? Well, in the light blue, yeah, you can see it there, good. In the light blue, that's the OP varieties. So they performed reasonably well. I think the CFX2 and rack were pretty early maturing, so we actually had to go and cut some of those sooner. But then some of the hybrids, they were okay, but some did exceptionally well. And you could tell just walking through the plots and looking at them, like this plot's going to yield extremely well. So this is maybe hard to read, but it's a summary of the mean compared to the check over on the right-hand side. So in the first column is the mean, on the right-hand side is the check. And almost the vast majority of those hybrids are within range, and I'd say about half of the ones there are, significantly better, like 1.5 to, in some cases, 2 times better. In some cases, some of the sites and locations, they were like 2.5 times better even. So there's some pretty interesting things happening. This is just coming on market. It's been available for quite a while within the CBD world, but it's just kind of starting to go into grain markets and things as well. So something to look forward to. And I think it's been pretty interesting to kind of figure out and learn about. One note, hemps are not automatically exempted from THC testing, but that's for a different time. So my main message here, grow hemp. I did doctor that photo a little bit. Maybe somebody will show you the real one at some point. How much time do I have? 5 minutes? 5 minutes. That went a little faster than I thought. So maybe I'll just quickly share the last little thing we're working on then. And then if there's time for any questions, we'll do some questions. So this other study that we're working on, this is actually our 2025 field getting prepped for next year. You can see we got really, really tall stubble. And this is looking at agronomic and chemical measures to improve germination and survivability. So those two things are pretty huge. We get like 30% emergence on some of our hemp year to year. So it's really not great. And if we're starting to get into hybrid seed and reducing seeding rates and things like that, we really need to dial that in and get better emergence from our hemp crop. So this is broken up into reps. One of the first things we're looking at is that stubble height. So we actually went back and forth in here, mowed a bunch of those blocks down so that we have some shorter stubble. And then we're planting two different varieties. So this Salacia is that fiber variety, and we're going to plant just at the higher traditional rate and then rack that grain variety. And so this is more of a grain rate in there. And we're seeing a few different things. So this is just some combined data from this year. So one of the interesting things we're looking at is the emergence counts and then the final plant stands. And so, hemp will have die-off from just self-thinning. It'll have problems with pythium that I'll show you here in a second. There's not a big difference between that regular and tall stubble, between the emergence and the final stand. We do see a reduction in both cases. And I wouldn't say there was any real difference here because of the stubble height. So our stubble wasn't, I wouldn't say, affecting our plant stands or our final stands. This is okay. So this is a Pythian slide. This is one of the main pathogens of hemp plants. It infects the root, and over on the left side there, you can see it's called an oomycete, and this was formerly categorized as a fungi. I mean, technically now it falls into this stramenophile category, which includes algae and other things like that. But we still kind of lump it all in with that just to make things easy, I think, a lot of times. And the thin brown roots there, those have been infected. The big white glowing plump ones are obviously uninfected. And you can see on the right side what happens. Eventually the hemp it'll just damp off is what it's called. So there's a number of really easy ways to deal with this. The first one is basically use seed treatment. This isn't something that's registered within hemp, so this will need an entire minor use and all sorts of things. But we're looking at metalaxyl M on this. That's the main mechanism for controlling oomycetes, pythium species, root rots, and other downy mildew types things. So we've got that within this study. And then this is just kind of a shot of us, initially attempting to treat that seed. Hemp's got a really kind of oily coating on it. And so with these water-based treatments, we had to refine things quite a bit, use, more misting applications, really low, what do you call it? Low spinning or kind of tumbling so we don't damage the seed. And so we spread things across really good. So we'll be looking at a combination, of all sorts of things here. So the metalaxyl on its own, metalaxyl plus fludeoxonil, and then also with the addition of sidaxane. So these are just commercially available seed treatments. Metalaxyl is like I already showed you there. And then the fludeoxonil and sidaxane will also target things like fusarium and rhizoctonia species and things like that. So yeah, a little tricky to get it all figured out. But once we did, we're seeing kind of some no results so far. So we've got some emergence and final stands. Is there a significant difference? I wouldn't say anything yet. Maybe some trends upward, so maybe some things to be like, hey, yeah, these seed treatments might be improving things, but it's really hard to say for sure at this point. And then on the fiber side, we didn't see anything this year yet. And I really don't expect to see probably yield effects from this, but definitely on the emergence. So hopefully when we get some more data, we'll see a little more positive trend there. And then, like I said, there's this entire minor use side of things to get registration ongoing. The biggest challenge is that a lot of the manufacturers don't necessarily want to support hemp. I think it's, this whole system is pretty backlogged, and who knows, it might not even be there a week from now. But this comes from the IR4 project. This is within the federal government. Everything's got to go through here. It's a 5-year review. And so if anybody is interested, I was kind of playing around with this site, and it will show you all the research that's been done for any product registration within North America. So it's a pretty powerful database of things that we have there. And that's what I got. That's what's happening. I hope my presentation was exemplary. All right.
Anyone with questions?
You can always catch me at the break if you got any burning hemp questions.
Well, thanks for that, Mike.
Yep.
So I think my terminology now includes mom's purple brox and dad's disease package to include that. So. Thank you, Mike.